recording. Okay, so physiology of the renal system. The picture in front of you, if you still remember the lecture of the gastrointestinal system, which we talked about the barium meal and the barium enema. This is something similar, a contrast study, which is given usually intravenously, and then it is used to visualize the urinary system. You can see from above the calyces and the pelvis of the kidney on both sides, and the ureter down to the urinary bladder. So this also can help to visualize if there is any obstruction due to a mass or due to a stone. And in case there is renal failure, it is also going to help. So the organs which you have been introduced to, the kidneys, the ureter. We have two ureters, two kidneys, urinary bladder, and the urethra. The kidneys are from all the parts of the renal system, the only one which is functional in forming urine is the kidney, while the rest aims only to transport and store the urine before it is released by urination or micturition. So even in the kidney itself, most of the structural units are present in the cortex. I'm sure you have been introduced to this. So the renal cortex is the part which contains all of the functional units known as the nephrons or the glomeruli, while in the medulla and deeper it's more in containing the tubular system, and we will explain why is that and how it helps and for what function. Okay, so the functions of the kidney in general or the renal system. Number one is removal of waste products. So as we all know that the human body has a lot of metabolic processes, and there are a lot of waste products produced within the body in the normal processes, including the urea, which is the end product of protein catabolism, creatinine, which is released by the muscles metabolism, uric acid, which is released from the nucleotide catabolism. So these waste products are harmful to the body if they are accumulated. Therefore, they are usually removed by the body into the urine with the help of the kidney. The kidney not only excretes the waste product, but it also reabsorbs the beneficial substances. There are some substances which will be filtered. So the kidney is actually consists of small millions of filtering units. So in that filtration, sometimes useful substances are filtered, and they're going to be lost into the urine unless they are recovered back, and that process of recovery is reabsorption. So in reabsorption, there are some vital or beneficial substances such as glucose. So glucose, and 100% of the glucose which is filtered, it is filtered. It doesn't stay in the blood. So it passes with the filtrate, but all of it is reabsorbed back. In addition to the glucose, we have amino acids. Also, they are totally reabsorbed back to the system because they are necessary. Bicarbonates, sodium chloride, phosphates, and water. These, of course, are regulated and reabsorbed based on the need. The kidney can also regulate, increasing their reabsorption when they are deficient, reducing their reabsorption when there is an overload. So that's why it is important also in maintenance of the homeostasis of most of the electrolytes, sodium, potassium, calcium, magnesium, chloride, bicarbonate. All of these electrolytes are regulated by the kidney. In addition, the pH, the acidity of the human body, which is around 7.4, it is partly and to large extent regulated by the function of the kidney. We will explain this function in more details at the end of the lecture. In addition, the kidney helps to control the blood volume. If you still remember, in the endocrine system, there was one hormone known as the antidiuretic hormone, or the vasopressin. That hormone helps to regulate the blood volume. In addition, we have another enzyme known as the renin, and the renin-angiotensin system that also is housed by the kidney, helps to regulate the blood pressure. And you have not been introduced to cardiovascular system. In the cardiovascular system, when we explain the regulatory mechanisms that regulate blood pressure, the kidney is one of the most important long-term regulators of blood pressure, controlling the volume and the resistance on the vascular system. So in addition, the kidney releases an important hormone which is known as erythropoietin. So erythropoietin is the hormone that enhances the formation of red blood cells. If anyone is going to donate blood, then the reduction of hemoglobin will stimulate the kidney to release the erythropoietin hormone. This hormone will stimulate erythropoiesis in the bone marrows, making more blood cells into the bloodstream. Prostaglandins, immunoglobulins are also released by the kidney. Renin, as we just mentioned, which controls the blood pressure. One important function is activation of vitamin D, which greatly helps in the homeostasis of calcium. So vitamin D, as we all know, is very important for the maintenance and especially the absorption of calcium, and it is also regulated by the kidney. So the kidney could help calcium homeostasis by directly affecting the rate of excretion or reabsorption of calcium, and by the activation of vitamin D. In metabolism, kidney with liver both are important locations for gluconeogenesis. In biochemistry, gluconeogenesis is the process of formation of glucose from non-carbohydrate substrates. This function is going to be primarily active among all of us, especially during Ramadan. So when we are fasting, and we especially after the first few days of Ramadan, because usually in the first few days of fasting, we will depend on our storage of glycogen, which is a storage form of glucose. After those few days have passed, then the body will start converting proteins and fats into glucose, and that process is gluconeogenesis, formation of glucose from non-carbohydrate substrates, and this is primarily by the liver, and also to lesser extent is a function of the kidney. So these are the basic functions of the kidney. So looking into the basic structure, I'm not going to talk about the full details because I'm sure you have been introduced in the last lecture in the anatomy about what are the nephrons and what are they composed of, but we will go very fast. As you can see, each nephron, the nephron first of all is the basic functional unit of the kidney, and these units are primarily situated in the cortex and in the corticomedullary junction. All of the glomeruli, which are the renal corpuscle, as you can see, the rounded shaped structure, which consists of a tuft of capillaries, a special type of capillaries into the Bowman capsule. That combination of Bowman capsule and the tuft of glomerular capillaries is known as the renal corpuscle. So that's one part. The second part is the renal tubules, which consist of the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. So all corpuscles are located in the cortex of the kidney. None are existing in the medulla. One of the reasons is that the medulla has a very high osmolarity. If you've been introduced to the osmolarity, which is how much solutes are present in our body. If you into all of the tissues in our body, you all have the osmolarity of 300 milliosmol, which is the same as what's inside the cells, but we have the only exception for this level of osmolarity is within the medulla of the kidney. It is very high, and it is very high for a reason that will be explained later. So this is the basic functional unit structure, the nephron. So this is the corpuscle again, and you can see the tuft of the glomerular capillaries, and covered by the podocytes, which are specialized cells with food processes that make slit-like openings, and this will control the filter that's going to be important in the process of excretion. Now, all of these capillaries, which are known as the glomeruli, are different from other capillaries. They are more permeable. If we know that we have capillaries all over our body, because in the capillary, that's usually how oxygen is given, carbon dioxide, but this capillary is different. It's not for that function. It has a different function, which is the formation of filtrate. They are very permeable, and they have also a higher blood pressure. In ordinary capillaries, roughly the pressure is around 14 millimeter of mercury, while here in the glomerulus it reaches up to 50 millimeter of mercury. So the pressure here is high. That helps in more filtration. In addition, these capillaries are also known as fenestrated capillaries, which has bigger opening that allows the movement of more substances to be filtered. But of course, in this filtration slits, not all particles can pass. Only the plasma is able to pass, but not all of the plasma. The proteins will not be passing through. The cells, everything will remain in the circulation. So only the liquid, the part of the plasma without the protein, is going to be filtered out from the glomerulus into the tubular system. And within that filtrate comes some substances which are important for the body, as we mentioned before, the glucose, the amino acids, so these will be reabsorbed back into the human body. So this glomerulus is the special filtration functional unit within the kidneys. How much nephrons do we have in each kidney? Nearly around 1.5 million in each kidney. And it consists of an afferent coming into the glomerulus and an efferent. The afferent branch comes from the radial cortical artery, and the efferent leaving the glomerulus will be forming another capillary network known as the peritubular capillaries. So as you can see here, this is another diagram of the nephron, shows you the glomerulus and the glomerular tuft of capillaries, and the pressure here is very high compared with the peritubular capillaries, which is the other network. So within the nephron, we have two capillary networks. One of them is the glomerulus, which has arteries on either side, while the other one, which is the peritubular capillary, it has an artery on one side and the vein on the other side, like most of other capillaries. Again, the glomeruli has higher pressure, more permeability due to their special structure, and surface area is also increased here. The process of filtration within the glomerulus is different from most of the capillaries, determined by the size of the openings within the fenestria. There is an additional function of the negative charge that will repel other negatively charged substances such as proteins. So this also helps to prevent proteins to pass through the glomerulus into the urine. Okay, so again, this is the structure of the nephron. As you can see, there is one additional part which I have not explained here. It is the juxtamedullary or juxtaglomerular apparatus, which is formed by the union of specialized cells within the distal convoluted tubule and the afferent arteriole, making that special system which release renin. And as we mentioned before, renin regulates the blood pressure, and it is primarily secreted from here. So the glomerulus is fed by the afferent, and then the artery which leaves the glomerulus is the efferent. The efferent comes from the cortical radiate artery, and then the efferent will feed another capillary network known as the peritubular capillaries. And this glomerulus is specialized for filtration, and it has more permeability compared with the usual capillaries that are present in our body. Filtration is not the only process that happens in the formation of urine. So if the question comes, and then whatever is filtered from the glomerulus, is it equal to urine? The answer is no. Urine is very different from what is filtered in the glomerulus. That's for one reason, because still there are a lot of modification that will be happening here. One of the modification is there will be some reabsorption. So reabsorption, some useful substances, as we mentioned, will be back into the circulation, into the peritubular capillaries, the other capillary network that we have just described. Sometimes there are some harmful substances which were not harmful, but maybe they are in high concentration, or there are some additional waste products which are not completely filtered, so those will be additionally secreted into the tubular system. So that's why the urine is going to be more modified on its way by the secretion and the reabsorption. So as you can see, glomerular filtration, here the water and solutes smaller than proteins are forced through the capillaries walls and pores of the glomerular capsule into the renal tubule. So this is a simple filtration. Of course, this process depends on the differences between the hydrostatic and agotic pressure. I'm going to explain this shortly. Basically, the hydrostatic pressure, if you remember, is the pressure which pushes. If you have two sides of a membrane, usually substance will move from high pressure to lower pressure, and that pressure is known as hydrostatic pressure. And it is the same here as the blood pressure, which is around, as we mentioned, 45 to 50 millimeter mercury. That pressure is also opposed by another pressure known as the ancotic pressure. So ancotic pressure function is slightly different. It is like a sponge. It doesn't push, it actually withdraws. It sucks like a sponge. So if we have those sponge-like forces within the capillaries, they will oppose the process of filtration. We will describe it shortly. So the glomerular filtration depends on the equilibrium, or the difference between the hydrostatic and ancotic pressures on either side of the filtration membrane. We have the tubular reabsorption, whereby substances which are useful are reabsorbed back. So these include glucose, amino acids, water, and usually this process of reabsorption, its majority, 60 to 70% of it, happens in the proximal convoluted tubule. So most of the reabsorption of all substances are within the proximal convoluted tubule. And tubular secretion, sometimes the hydrogen, potassium, creatinine, some drug metabolites, harmful waste products will be released into the tubule by the process of secretion. So secretion is something which is not completely present in the filtrate, and then is going to be released into the filtrate from the peritubular capillaries. So here this diagram shows you different substances, which are how they are reabsorbed and how they are secreted or filtrated. One important thing to be mentioned is that I just told you that all of the glomeruli are present within the cortex of the kidney, but there is a classification. Majority of the glomeruli or nephrons are known as cortical nephron, and these are 85%, and all of it, nearly all of that nephron is present within the cortex. We have another group of nephrons known as the juxtamedullary nephrons. Those nephrons are slightly, the glomerulus is still in the cortex, but the tubular system dives deeper into the medulla. And these 15% of nephrons create the hyperosmolar medium in the medulla of the kidney, which is very vital. Without it, we will be dying, because I didn't mention to you, but according to the glomerular filtration rate, which is around 125 ml of fluid per minute, if we try to make the calculation of if that all fluid ends up as urine, then we will be releasing 180 liter of urine every day. So that is impossible to survive. But we have this very special and useful hyperosmolar environment in the kidney that reabsorbs most of the water, which is filtrated, and give it back into the human body, and makes the body able to survive even in hot environments. And I will explain to you what's the difference that happens while we are fasting, and how these juxtamedullary nephrons help to protect us by creating the special hyperosmolar environment within the medulla of the kidney. Okay? So 15% of nephrons are juxtamedullary nephrons. The majority are cortical, which are present with the corpuscle, and the tubules are within the cortex, while juxtamedullary, the corpuscle and the convoluted tubules are in the corticomedullary junction, while the loop of Henle dives deeper into the medulla, creating that hyperosmolar environment. So here again, the two classes of nephrons are shown in this figure, the cortical and the juxtamedullary, which has a longer loop that dives. Even the shape of the vessels, the peritubular capillaries, are different here in the juxtamedullary nephrons. They are known as the vasa recta, which also helps to maintain the function in the medulla of the nephron. Okay, so here the renal corpuscle is shown again. So you can see the Bowman capsule, the tuft of capillaries, the juxtaglomerular cells, the macula densa, the afferent arteriole, the efferent arteriole, and how this system helps to release the renin that helps to regulate the blood pressure in the human body. So the renin-angiotensin mechanism, which is just explained to you, consists of the JJ cells, which are present near to the afferent arterioles, secreting the renin that helps in the activation of angiotensin II and its formation by the angiotensin converting enzyme. This end product will stimulate the secretion of aldosterone, and it also enhances the vasoconstriction, which is very important to maintain the blood pressure. So renin-angiotensin mechanism is actually activated when the blood pressure is low. So when the blood pressure is low and there is low solutes, this will be sensed by the kidney juxtaglomerular cells. Now, when they have sensed that low solute, low blood pressure, then they will start release the renin enzyme that will start the chain of reactions, ending up with the formation of angiotensin II. First of all, angiotensinogen is going to be released by the liver, and by the renin is going to be converted into angiotensin 1. Then angiotensin 1 will be converted into angiotensin 2 by the angiotensin converting enzyme released by the lung. And this angiotensin 2 will have many effects. First of all, it will stimulate thirst, so you will feel thirst, which is also helped by the osmoreceptors present in the thalamus. And angiotensin 2 will start enhance the immediate vasoconstriction, increasing the peripheral resistance, leading into an increase in the blood pressure. And also it stimulates the release of aldosterone from the adrenal cortex. So the aldosterone enhances the reabsorption of sodium and water, and this will altogether help to increase the blood pressure. Okay, so the renin angiotensin aldosterone system is shown here in the diagram. As I just mentioned to you, the liver, the kidney, and the lung. So the end product, which is angiotensin 2, helps in vasoconstriction, releasing aldosterone, in addition to the stimulation of thirst and, to a lesser extent, the activation of antidiuretic hormone. Antidiuretic hormone is more stimulated by the osmoreceptors, which are present in the thalamus. So here in this diagram, you just look onto the left side. In the left side, when there is a fall in the blood pressure, this will stimulate the JJ cells of the kidney, the juxtaglomerular cells. So they will sense the reduction of blood pressure. They will sense the reduction of solute content in the renal tubule. So renin will be released into the blood, and the presence of renin will end up with the formation of angiotensin 2 in the blood. That will have a direct effect on the arteries and arterioles, causing their vasoconstriction, increasing the resistance. That will lead to an increase in the blood pressure. At the same time, the angiotensin 2 stimulates the adrenal cortex, as we mentioned, releasing aldosterone that will help in the reabsorption of sodium with water in the distal convoluted tubule. That will increase the blood volume, and again, it will increase the blood pressure. So this is one of the mechanisms which controls the blood pressure. The other one, which is in purple here, is the hypothalamic osmoreceptors that will also sense the fall in blood pressure. So here these osmoreceptors will release the antidiuretic hormone, as I mentioned to you, that will help in the reabsorption of water. So this is what happens while you are fasting in Ramadan, especially towards the sunset. So if you are dehydrated, then your body senses that your osmoreceptors will sense this dehydration, and you will feel thirsty. At the same time, the hypothalamus will release the antidiuretic hormone through the posterior pituitary gland into the circulation, enhancing the more reabsorption of water by the collecting ducts, making the urine very concentrated and in very little volumes. This is mainly due to the less amount of water. So the glomerular filtration rate, which is the rate of filtration through the glomerulus, the volume of fluid filtered by the glomerular membrane of the kidney per minute. And that amount is 125 ml per minute, as I mentioned before. If we make a calculation per day, it's nearly 180 liters per day. So how can we survive? Our vascular system, or the blood, is only 5 liters. So how are we able to form 180 liters per day? That's because we are 99.9% we are reabsorbing it back into the circulation, and a small portion of it, which is approximately 1.5 liters, is released every day as urine. So one thing important to remember, how the function in the glomerulus is regulated. I just told you about the hydrostatic pressure and the oncotic pressure. So hydrostatic, so you can imagine this diagram here. It shows you on the left side an afferent arteriole. From here, bloods come into the glomerulus. And then we have the efferent from where the blood is leaving the glomerulus into the peritubular capillaries. So here, this is the filtration membrane. And down here is the tubular system. Okay? So we have different pressures working on either side of this filtration membrane. We have hydrostatic pressure, which is the PGC. So the more hydrostatic pressure here, this will make more filtration to pass to the tubules. We have another hydrostatic pressure in the tubular system, which is known as PT. This is going to oppose the filtration because it's going to push in opposite direction. So if this pressure increases, this will reduce the amount of filtration. We have the ancotic pressure here, the delta GC or kappa, this symbol. So this kappa GC is ancotic pressure. And as I mentioned to you, ancotic pressure works as a sponge. So the more ancotic pressure here, again, this will suck more fluid from the tubular system back into the tuft of capillaries. So this will oppose the process of filtration. So the ancotic pressure is largely determined by the plasma proteins. So plasma proteins here are going to exert an ancotic pressure that will reduce the amount of filtration. So in people who have certain diseases in which there will be low level of plasma proteins, then their GFR or glomerular filtration rate will be more because there will be less ancotic pressure and the hydrostatic pressure is going to be unopposed and is going to push more. Even worse, if the problem is in the kidney, causing the plasma proteins to be lost into the filtrate, so this will increase the ancotic pressure on this side, which will increase the GFR. And that's why in certain people with renal disease, they have large volumes of urine formation, large volumes due to the increase of filtration. So the infiltration will be positively related to the hydrostatic pressure in the glomerular artery capillary, okay? And it is negatively related to the hydrostatic pressure in the tubular side. It is negatively correlated with the ancotic pressure in the glomerular capillary, which is indicated by the level of plasma proteins. So this equation here shows you the difference between the two hydrostatic pressure minus the difference in the ancotic pressure equals the GFR if we multiply it by a constant or filtration coefficient. So you're not going to do any calculation, but this just helps you to understand the equation and what are the factors that controls the glomerular filtration rate. So the coefficient determine, it also depends on the permeability, which is also modifiable in certain diseases or in age, can also affect the permeability. Hydrostatic pressure in the glomerulus could also be affected in people with, for example, hypertension or with factors that could affect the renal blood flow. And the hydrostatic pressure in the tubule, which is in the filtration tubule, this is only going to be changed if there is an obstruction in the urinary system, causing backflow of urine, increasing this hydrostatic pressure, as happens in the stones, tumors. Even in pregnancy, the uterus could press on the ureters, making a backflow of urine that could increase the hydrostatic pressure in the tubules. While the ancotic pressure, as we mentioned, the sponge action, the ancotic pressure here of the plasma, which is by the plasma proteins, while the ancotic pressure in the tubule is zero because normally there is no protein that is filtered in the tubules. That's why it is zero. But in renal diseases, if there is damage making the proteins lost into the urine, then it will be counted. It will be counted and it's going to be affecting the GFR. So GFR here again, the filtration coefficient and the same factors that I have explained. So the hydrostatic pressure in the glomerulus, we just mentioned before, is determined by the blood pressure in the glomerular capillaries. So if there is hypertension, this could increase the glomerular filtration rate by increasing the hydrostatic pressure. Sometimes there is also a difference in the degree of constriction and dilation between the afferent and efferent arterioles of the glomerulus. So you can imagine if the afferent is dilated while the efferent is constricted, this will make the pressure higher in the glomerulus and it will make more hydrostatic pressure. While if the opposite happen and the afferent is constricted and the efferent is dilated, then the pressure will be low. So these changes in between afferent and efferent could happen by different neurotransmitters such as noradrenaline and also by angiotensin, which is one of the important regulator. It is a vasoconstrictor, but it has more vasoconstriction on efferent than afferent. So the afferent will be more dilated, increasing the flow, and the efferent will be squeezing the blood, making less blood passing into the peritubular capillaries, and this will increase the pressure more in the glomerulus, making more hydrostatic difference and more filtration happen by the effect of angiotensin 2. So angiotensin 2 increases the GFR, noradrenaline increases the GFR, hypertension increases the GFR. So here, if there is any increase in the renal blood flow, it can also lead to an increase in the glomerular filtration rate. So, but here, if there is a constriction in the afferent, this will reduce GFR. If there is constriction in the efferent, this will increase the GFR, as I just mentioned to you right now. So the hydrostatic pressure in the tubular system, normally nothing can change it because it's just urine is formed and it's released to the bladder, and the bladder is releasing the urine through micturition, and that's constant. But it could be increased if there is an obstruction, such as a stone or a gravid uterus in pregnancy, or edema due to some inflammation that will press on the renal tubules. So this will act against the glomerular filtration, reducing the glomerular filtration rate. So these are the two. I think we will reach next to the oncotic pressure. So these are the two factors that could change the hydrostatic pressure in the tubules, the gravid uterus and the stones in the renal system. Oncotic pressure, which is affected by the plasma proteins. And here, if there is hypoproteinemia, as we mentioned, if there is such as in liver disease, hypoproteinemia, this could reduce the oncotic pressure. So there will be less sponge activity in the glomerulus, and this will make GFR more. While in dehydration, the plasma proteins concentration will increase, and this will reduce the GFR. So these are basically the factors, while the oncotic pressure here is negligible, 0. Unless there is protein loss in urine in kidney disease, then it will be counting. So these are the factors that could affect the glomerular filtration rate. So this is summary of all of it together. Changes in renal blood flow. So an increase in renal blood flow makes an increase in the glomerular filtration rate. The changes of constriction between the afferent and the efferent, as we just mentioned, could affect the GFR. Hypertension can also lead to an increase in the GFR. Certain neurotransmitters and noradrenaline, angiotensin 2 can affect the GFR. Pressure in the tubular system by a stone or a pregnancy, as we mentioned, can also affect the GFR, reduce it. So number 1 and 2 increases the GFR. Number 3 decreases the GFR. Number 4 changes in the concentration of plasma proteins can also, if it is increased, it will decrease the GFR, such as in dehydration. While if there is liver disease and there is hypoproteinemia, then this will increase the GFR due to the reduction of the oncotic pressure. Changes in the coefficient, which is the changes of within age that affects the surface area. And 5 and 6 are less important, which is the contraction of the mesangial cells, which is also influenced by the angiotensin and vasopressin. You just need to focus on the first 4 factors. So now we have reached to the important topic of regulation of water homeostasis by the kidney. So how does the kidney work into the regulation? So I think by now we understand what is the basic functional unit, what is the structure of it. We have talked about the juxtaglomerular apparatus that releases renin, which regulates the blood pressure. We talked about the filtration through the glomerulus and the GFR and the amount of it and how it changes and what regulates it. The different functions are the factors that could be regulating the GFR. In Arabic, oncotic pressure, I'm not sure, but it is the pressure exerted by the proteins which are available within the plasma. So plasma proteins are the main contributor regulating the oncotic pressure. And its function, to understand it, it's like a sponge. So it absorbs the fluid. So the more plasma proteins in your blood, this will take the filtration back into the blood. So it works against the filtration. So regulation of water or homeostasis by the kidney, which is an important topic. So first of all, we need to introduce, in general, the water is regulated between how much we take and how much we lose. So normally the intake is distributed between food and drink into a certain amount. Of course, this is not fixed, but if we are talking about an average value, 2,300 ml. So the amount of losses, of course, we lose through the skin, insensible. This is not sweating. The skin is always kept moist by this insensible loss. In addition, there is some insensible loss by the ventilation through the lungs. Then there is loss through sweating, and this is modifiable, such as in heavy exercise. As you can see, the 100 ml loss will become 5 liter. So when you are running or in very hot environment, then there are a lot of fluid will be lost through sweating. Certain amount is lost in feces, and then usually what is left is going to be lost in urine. So for example, in hot environment, in prolonged exercise, if we are losing a lot in sweating, then the urine volume will become much lower. And the opposite, in case you are drinking a lot of fluids and you are overhydrating yourself, like what happens before imsak, during the beginning of the fasting day. So in the morning, then you'll be passing large volumes of urine because there is an overload of fluid, and of course this will not remain in the circulation. The body will lose it and not reabsorb it back, and it will be lost in urine. Of course, that amount of intake and loss is regulating directly the plasma, and that plasma is around 3 liter. The blood volume is 5 liter. If we take out the cellular part, which is 2 liter roughly, the plasma is around 3 liters. So usually the losses and the intake directly contributes to that 3 liter. But we have this 3 liter is in direct exchange with the interstitial fluid through the capillary membrane, and that interstitial fluid volume is around 11 liter, while the majority of the fluids in our body, which is the intracellular fluid, around 28 liter, is in exchange with the interstitial fluid. So that's how, if there is any problem, it's going to change stage by stage from intracellular to interstitial, then it will be reached into the plasma. And as we mentioned, these are the organs responsible for the losses. So the kidney is one of the important top of the list organs responsible for loss of water. But this loss, as we know, is regulated. When we are dehydrated, it's going to be less. When we are overhydrated, it's going to be more. Okay, so it's not as a fixed value. Otherwise, it will be impossible to survive. So we have, of course, mechanisms related to intake of water, and that mechanism is the thirst mechanism. Now, if you are thirsty, which is happening due to the stimulation by the osmoreceptors in the hypothalamus, so these osmoreceptors will sense the osmolarity of your blood. If it is increased by the increased concentration of the solutes, then you will need to take more fluid. Then you will feel thirst by their stimulation. So they're going to be active, and then when they're active, then the hypothalamus, of course, will undergo a number of responses. One of them, the hypothalamus will enhance the reabsorption of water from the kidney by the antidiuretic hormone. It also stimulates thirst, so you will drink, and this sensation is also, when the mouth is dry, it can also make you feel thirsty. So here, an increase in the plasma solutes, which means dehydration, it reduces the saliva and it leads to a dry mouth. So dry mouth can also stimulate the hypothalamic thirst center. And the osmoreceptors also in the hypothalamus will be stimulating the thirst center. So you will feel thirsty, and then this will stimulate you to drink fluids, and this sensation of thirst will enhance drinking, and with drinking, the hydration of the oral cavity and distension of the stomach and hydration of the throat can also relieve that sensation and stop the stimulation of the thirst center. And of course, after absorption of water, which takes some time, this will reduce the osmolarity of the plasma, bringing it back to normal. So this will interfere with the cycle through the negative feedback mechanism. Which values you want to remember? Do you need to memorize all these values? Which values? So you're talking about this figure? Previous slides. I'm not looking actually. So maybe you can ask me at the end, okay? So just take note, and then we will give some time at the end. You can ask. Okay, so the urine concentrating power, as we mentioned that in the glomerular filtration, we nearly make 180 liters per day, but what ends up as urine is roughly half to 1.5 liters per day. So we are reabsorbing nearly 99, even more than 99% of the water, reabsorbing it back. So how does that work? This, of course, by a special system, by the special nephrons that I have explained to you in the beginning of this lecture. I told you about a certain group, 15% of the nephron known as the juxtamedullary nephrons. These nephrons, with their special loop of Henle, which is diving deeper into the medulla, trapping, they work as a special function of pumping solutes, pumping urea, increasing the solutes within the medulla, making this medullary environment hyperosmolar. So I don't want to take you into the details because it is step by step and slightly complicated topic to be understood. Just remember that the hyperosmolarity of the medulla of the kidney is created by the loop of Henle of the juxtamedullary nephrons. And these juxtamedullary nephrons, or the loop of Henle, will work as a countercurrent multiplier, multiplying the osmolarity within the medulla, trapping the solutes within there. And at the same time, we have a countercurrent exchanger, which is the vasa recta, which is the peritubular capillaries that will wash out the water and prevent the dilution of the solutes within the medulla. So in that way, the water will always be washed out from the medulla and the solutes will remain there, making it very hyperosmolar. So the countercurrent system consists of the loop of Henle and the vasa recta. These two systems are vital for the hyperosmolarity. The loop of Henle will work as a countercurrent multiplier, increasing the medullary hypertonicity, while the vasa recta will maintain it by washing out the fluid. And these two systems, without them, we will not have the hypertonic medulla of the kidney. And without the hypertonic medulla of the kidney will lose its concentrating ability, and if we lose it, then we will die. We will not be able to survive. The body will be dehydrating within few minutes to few hours. All of the fluid in the plasma will be lost. Okay, so this is the, as you can see in the diagram, the osmolarity from the cortex 300, which is similar to the intravascular osmolarity, but then suddenly it increases in the medulla up to 1200, which is a very high value, and this value is important when we are dehydrated. So normally what happens is that the urine is formed in the glomeruli, and this urine will be passing into the tubules that will pass from the cortex into the medulla, uniting among each other to collect or divert the urine into the minor calyces and major calyces, later into the pelvis of the kidney. But on that way, if we are dehydrated, then there will be a release of antidiuretic hormone. The antidiuretic hormone will make the tubules permeable to the environment. So what happens? The fluid will immediately leave the tubules, the collecting ducts, and all of it will be passing into the environment, which is hypertonic because it's of high salt content, making a very efficient absorbent of fluid which is there. It will not allow the fluid to pass outside as urine. It will take it all from the urine and making the urine very concentrated. And this will work only when we are dehydrated. Now, any one of you can think, while we are well hydrated, why doesn't that work? Because we are always having this hyperosmolar environment. So what's the difference when we are overhydrated? Why does the urine not become concentrated? Why is it diluted when we are well hydrated? Any answer from your side? Yes, but why? Water is reabsorbed? Why? How is water? When we are well hydrated, of course, water reabsorption is distributed on different parts of the nephron. Majority of water reabsorption happens in the proximal convoluted tubule, part of it in the distal convoluted tubule. Just think about it in this way. You have this environment in the kidney, which is hyperosmolar in the medulla. And this hyperosmolarity is constant. It never changes. And you have the tubules of the nephrons all passing through this environment. But when you are well hydrated and you don't need to reabsorb it, the tubules are not going to be permeable. So the fluid will remain inside and there is no contact with the environment. And the fluid will end up as urine. But when you are dehydrated, your body needs that fluid. What happens? There will be a release of the hormone known as the antidiuretic hormone. That hormone, it will function on these collecting ducts and make them permeable. So suddenly there will be windows and doors open to the environment around these collecting ducts and tubules. And that will absorb most of the fluid which is inside the tubular system. And that's how the urine become concentrated. So I hope this is clear now. And I think there is another slide that shows you the function of the antidiuretic hormone. So it is important that the hyperosmolarity in the medulla is created by the loop of Henle, which works as the countercurrent multiplier, while it is maintained by the vasa recta, which is the peritubular capillaries working as the exchanger that washes out all of the water, preventing its trapment in the medullary interstitium, making it and maintaining it as a hyperosmolar. So this is the countercurrent system, which consists of the loop of Henle and the vasa recta only in the juxtamedullary nephrons, not the cortical nephrons. This is 15% of the nephrons which are present at the corticomedullary junction, and their loops are dive deep into the medulla, creating this environment. So if the collecting duct is permeable to water, which means this is with the presence of vasopressin or antidiuretic hormone. If there is no vasopressin, then it is not permeable, and all of the fluid will be lost as urine. So this is what's going to happen when you are well hydrated or dehydrated. This is the factor that will determine how urine is going to be. So this figure here shows you on the left side, this is a well hydrated person. So maybe this is the condition of us in Fajr, before sunrise, when we have already drunk a lot of fluid in before we stop eating and drinking. So what will happen? There is a lot of fluid there. The body will not release antidiuretic hormone. So the collecting ducts will be not permeable to the hyperosmolar medulla, and most of the fluid will end up as large volumes of urine, dilute urine. But what happens toward the end of the day before we break fast, the body is dehydrated, so antidiuretic hormone will be released. The antidiuretic hormone will make the collecting ducts permeable to the environment. So all of the water, or most of it, is going to be leaving, making the urine concentrated and in small volume. So this takes us to the next slide about urine. So urine, so we finished now the topic of hyperosmolarity. So the volume of urine is around 1.5 liter per day. Is urine and filtrate the same? Of course, it's not the same, because we have plasma proteins, we have this, is the urine and filtrate the same? No, it's not the same, because of the reabsorption and secretion, as we mentioned before. So there are some substances which are added, there are other substances which are taken back. So this will change the composition of the filtrate, and it's not going to be the same as urine. So the characteristic of urine, it's yellow amber color due to the urochrome pigment, which comes from the catabolism of heme, which is part of the hemoglobin. The pH is slightly acidic normally, but this of course varies. The kidney, as we mentioned in the beginning, function also as in maintaining pH. So if our pH is more acidic, the kidney will lose more acid and it will become more acidic. If we are alkaline, then the kidney will become alkaline, releasing more alkaline into the urine to compensate and make the pH back to normal. It is also having a smell due to the ammonia. Okay. Solutes which are normally found in urine. Now this is important. What is possible to be found in urine? Sodium and potassium, okay, it's acceptable to find them. Urea, end product of protein catabolism, uric acid, creatinine, ammonia, bicarbonate, these are acceptable to be found in the urine. What is not acceptable? Now, if anyone has a problem and then they will take a sample of urine for general urine analysis, what is acceptable and what is not acceptable? Now, those which are in front of you are acceptable to be seen in the urine sample. But what is not acceptable to be seen? Glucose. Now, if there is any glucose in urine, this means the person is diabetic. And I will explain why does the glucose in diabetic patients appear in the urine. So glucose should not be present. It should not be detected in the urine. What else? Proteins. So no protein should be there. If there is any protein in the urine, this indicates renal disease. Red blood cells, which means there is bleeding, either a renal kidney injury or a stone in the ureter or urinary bladder, which is making injury, releasing blood cells into the urine. So no red blood cells, no hemoglobin, no white blood cells. So no red blood cells, no white blood cells, okay? And bile should not also be detected. So if bile is detected, this means the person is jaundiced, okay? White blood cells indicates there is an infection. Red blood cells indicate there is an injury. Hemoglobin indicates there might be hemolytic anemia. Plasma proteins indicate a kidney disease. Glucose indicates there could be diabetes mellitus. So here are those which I have just explained to you. So here I'm going to explain why does the glucose should not be present in the urine. As we mentioned before, is that glucose is filtered through the glomerulus, but all of it is reabsorbed. 100% is reabsorbed. So there should be no glucose in urine detected. So this is mainly due to the presence of transporters of glucose within the tubular system of the glomerulus. So once these proteins, which are carriers for glucose, are saturated, which means the load of glucose in the filtrate is too high, more than what they can carry, then the glucose will start appearing in urine. That's why it is diagnostic of diabetes, because in diabetes the blood glucose is very high. So all of that amount, it is impossible to reabsorb it all back to the circulation. Therefore, certain amount is going to be more than the capacity of the kidney, which is also known as the renal threshold. So the renal threshold of transporting the glucose to the circulation is going to be overcome by the high concentration of glucose in the blood. And then the glucose will start appearing in the urine in those diabetic patients. So it's not due to a renal disease, although renal disease happens in diabetes, but it is due to the having a load of glucose which is more than what the kidney can reabsorb back. So that's called the renal threshold, and it is mainly due to the transporters of glucose which are saturated and they are unable to reabsorb the remaining parts. So this is the renal threshold, which is around 180 milligram, which is equal to 10 millimole. So this is the level of glucose at which it might start appearing in urine. Okay? So now this is a new topic, micturition or urination. We will understand how urination is regulated and how does it work. So, so far, any question from your side? Give me just 1 minute before we go into it. What is the difference between the two types? Two types of what? The pressure becomes... What's the difference between the two types of nephron? Well, the two types of nephron, in structure, they are very similar. They both have the glomeruli, the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, okay? And both of them also work in filtration, okay? The formation of urine. But the difference is that in these juxtamedullary nephrons, their loop of Henle dives into the medulla and perform a special function only for them by pumping out the solutes, the sodium chloride, the urea, and trapping them in the medulla. So that's the difference. The cortical nephrons does not do that function. They only work in formation of filtrate, making urine, while those in the juxtamedullary junction, which is known as the juxtamedullary nephrons, they make the filtration and urine. In addition, they create the hyperosmolarity of the medulla, which is important for the reabsorption of water, as we explained before. Yeah. You're welcome. Okay, so the next topic is going to be about micturition or urination, voiding or emptying of the urinary bladder. So if you are still aware about what you have studied, I think you also been introduced to the micturition in the anatomy lecture by Dr. Hala, because I had looked into the slides. It's in the last slide. Anyway, we will talk about it more. So basically, there are two sphincters that control the urination from the urinary bladder. One is known as the internal sphincter, internal urethral sphincter, and one is the external. The internal is involuntary, which is primarily controlled by the sympathetic nerve fibers, and the external urethral sphincter, which is controlled by the pudendal somatic nerve, that is under voluntary control. The first feeling of urge starts when the urine volume becomes 200 ml. So the capacity of the urinary bladder is nearly half liter, but it's not gonna, the feeling or the need to urinate will not start when it is full. Usually the starting feeling will appear when the volume is 200 ml and more. So what happens is there are some stretch receptors within the wall of the urinary bladder that will send impulses to the sacral region of the spinal cord. So for your information, micturition is a spinal reflex, which means the reflex of sensing the full bladder and giving the order to evacuate it. It is all regulated at the level of the spinal cord without involving the higher mental function. But what happens is that there will be some influence by the cerebral cortex, and most importantly the pontine micturition center, that will have some additional control on this spinal reflex, which I'm going to explain. So the micturition reflex consists of a spinal reflex that is dominant in early childhood. So in childhood, and that's why we tend to use the diapers, because they have not yet developed the pontine micturition center, is not dominant in controlling urination. So it is basically controlled at the spinal reflex. So once the bladder is filled, then immediately the reflex will relax the sphincter and urination will happen. And thus, this of course requires training in children in order to make them train their pontine micturition center to stop this reflex and making the urination only happen when it is convenient. So what happens is that when the bladder is containing the amount of that stimulates the stretch receptors, so this will be carried out by the sensory nerves to the spinal cord at the sacral region, and then the parasympathetic fiber through the symplanchnic branches will stimulate contractions in the detrosal muscles of the urinary bladder. So the urinary bladder will contract. At the same time, the sympathetic fibers will relax the internal sphincter, and this will enhance the passage of urine into the urethra. Of course, if there is no mature pontine center, then immediately urine will be released outside and urination will happen. But the external sphincter, which is voluntary control, this is going to be controlled primarily by the pontine micturition center. So here, this diagram shows you what happens. So the reflex is regulated at the sacral region by the sensory and motor fibers of the parasympathetic and sympathetic nervous system, which work against each other. So when the sympathetic system is active, the fibers will constrict the sphincter, and at the same time the urinary bladder will be relaxed. While when the parasympathetic system is active, then the urinary bladder will contract and the sphincter will relax. Now, of course, there is an additional control by the pudendal nerve, which is somatic, voluntary, and this is going to be primarily controlled by the pontine micturition center, which again is under the control of the cerebral cortex. It is under our will, under our decision. When it is convenient to urinate, then the pontine micturition center will enhance the relaxation, so it will stop the inhibition of the reflex, and it will make the bladder contract, the sphincters relax, and urination happen. So of course this feeling of urgency will increase with the increasing volume. So with more, the starting feeling, as we mentioned, in 200 ml, and this will intensely increase with more volumes up to 500, which is the maximum amount that will cause the maximum urge to urinate. So these are the sphincters and the detrosal muscles, as we just mentioned. Again, this shows you the spinal reflex that controls urination at the sacral, lower lumbar and sacral region, controlling the internal and external sphincters. So the external sphincter, which is important. So here this diagram is important for you. So the detrosal muscle, which causes the contraction of the urinary bladder releasing urine, is under parasympathetic control. And during filling, it is inhibited. During micturition, it's stimulated. So the urine bladder will contract, releasing urine by this parasympathetic fiber, which is also known as the symplanchnic pelvic nerve fibers. The sympathetic fibers, which control the internal urethral sphincter, here they are stimulated during filling, so they will prevent passage of urine. They will be inhibited during micturition. External urethral sphincter, which is voluntary skeletal muscle, is under the pudendal nerve, which is a somatic neuron, and it is stimulated during filling, and it is inhibited like the internal sphincter during the micturition. So while in micturition, the sphincters will be inhibited, the bladder will be contracted, and the parasympathetic system will make urination happen. So one of the parasympathetic system effects is enhancement of urination. So this figure here shows you the urethra in male, mainly because of the presence of the prostate gland, which is only present in male. After the external urethral sphincter, this accessory organ of the reproductive system, which we are going to explain in next lecture, it also might be involved in certain diseases, as in hypertrophy in old age groups, that will cause an obstruction to the urine outflow. So I think the last part of the lecture is about the acid-base balance and how the kidney maintains the pH. So the pH is very important in homeostasis. Our body needs to maintain the pH at a constant range of homeostasis between 7.35 to 7.45, and this is very vital for the functions of the enzymes and the different cells in our body. So without a constant pH, all the systems will collapse. So the kidney is part of the regulatory mechanisms that controls the acid-base balance and controls the pH. So we have certain substances that helps to regulate the pH in the body, and these systems are known as buffers. So the buffers will bind with hydrogens when they are acidosis, release hydrogen when there is alkalosis. So they will help to moderate the level of the pH. Now, among these buffer systems, we have the bicarbonate buffer system, and we have the phosphate buffer system and the protein buffer system. The more important is the bicarbonate buffer system within the function of the kidney. So just understand it very easily, as the kidney has the function of regulating the pH in a way that when we are having acidosis, the kidney will reabsorb more bicarbonate, which is alkaline, and will release more hydrogen, which is acid. So the body will lose acid and reabsorb more alkaline. And the opposite will happen when there is alkalosis. So when there is alkalosis, then the kidney will tend to lose more bicarbonate, and more hydrogen ions will be reabsorbed, and in that, or less will be lost in urine. So this will put the pH back within the normal range. And of course, if you still remember the respiratory system, which we have explained before, we mentioned about its role in controlling the pH by controlling the level of CO2. So when we have more CO2 in our body, the CO2 will change into carbonic acid, and this will reduce the pH. And the respiratory system with the renal system together can work in controlling the pH. Respiratory system through ventilation, renal system through urine formation. So the bicarbonate system is the major extracellular buffer system, which is functional by the kidney through the formation of weak acids from strong acids. You don't have to bother yourself, as I just told you before, how the renal and respiratory system works together. I'm going to show you a diagram at the end, and I just want you to understand that diagram, because that diagram is very important to understand the acid-base balance. So the kidneys and respiratory system work together to regulate the hydrogen ion concentrations. And the renal mechanisms, as I told you, when the blood pH is high in alkalosis, then bicarbonate is going to be excreted, hydrogen is going to be retained, while when the blood pH is acidic, falls, bicarbonate will be reabsorbed, hydrogen is going to be lost. And that's how we have a urine pH between high acidic into an alkaline, which will be according to the pH of our body. So if the body is alkaline, then the urine is going to be alkaline, losing more alkaline. Okay? If we are acidotic, then the urine will become acidotic, losing more acid and keeping more alkaline to keep the pH within normal range. So this is the diagram. So usually in the acid-base balance, what you are going is to have a scenario and a results of analysis. And based on the results of analysis, you are supposed to understand what is the situation. So basically, in that scenario of the arterial blood samples, first of all, you need to look into the pH value. If you look into the pH value and that pH is more than 7.4, then straight away you know that this is alkalosis. If it is less, then straight away that's acidosis. Now, acidosis could be metabolic or respiratory. Alkalosis could be respiratory and metabolic as well. So let's take the first alkalosis. When we first found the pH high, we know it is alkalosis, but we need to know what type of alkalosis it is. So next, we will look into the PCO2. If the PCO2 is less than 40, if you still remember respiratory system, the normal PCO2 is 40 in the arterial blood and is 45 in the venous blood. So if it is less than 40, this means it is respiratory alkalosis, like what happens in panic. When a person is ventilating very fast, washing out the CO2, making the level very low, and making alkalosis. The same thing which we have described in those who tend to dive by hyperventilating to prolong their diving time, if you still remember in the respiratory system. So here, this is the first type. If the pH is high and the PCO2 is low, then this is respiratory. If the pH is high and the PCO2 is normal, okay, then we look into the bicarbonate level. If the bicarbonate level is high, more than 24 milliequivalent, then this is metabolic. So if CO2 is normal and the bicarbonate is high, then this is metabolic alkalosis, not respiratory. Respiratory alkalosis if the CO2 is low with the high pH. Okay? Let's go to the other side. When the pH is low and acidosis, if that acidosis is caused by respiratory system, which means the respiratory system is not ventilating and the CO2 levels are increasing, changing into acid. So when the PCO2 is high and there is low pH, then we know that this is respiratory acidosis. Okay, so we see the pH is low and the PCO2 is high, higher than 40. Then this is respiratory acidosis. But if the PCO2 is normal and the pH is low, so we look into the bicarbonate level. If it is lower than 24, then together we will know that this is metabolic acidosis. So first thing, we need to look to the pH to know whether it is alkalosis or acidosis. The second thing, we look into the CO2. So if it is alkalosis and the CO2 is low, then we know it is respiratory. If it is acidosis and the CO2 is high, then we know it is respiratory. If it is alkalosis and the CO2 is normal, while the bicarbonate is high, then we know it's metabolic. If it is acidosis and the PCO2 is normal and the bicarbonate is low, then we know this is metabolic acidosis. Now, in respiratory alkalosis, the compensation will be renal, which means the kidney will lose more bicarbonate. So you might find with the high PCO2, not a normal level of bicarbonate. If the normal level of bicarbonate is present, then you know it is uncompensated. But if it is compensated, then you will find low level of bicarbonate because the kidney is losing bicarbonate. So that's how you know it is compensated or uncompensated, by looking after the CO2 into the bicarbonate level. If it is normal, then it is uncompensated. If it is low, then it is compensated. If the alkalosis is metabolic, which means the PCO2 was normal, then you looked here into the HCO3 and you found it's high, then you know it is metabolic alkalosis. In this case, the compensation is going to be more respiratory. So the respiratory system will reduce the alkalosis by trapping more CO2 by hypoventilation. So alkalosis, if it is metabolic, the respiratory compensation, how do we know it? Instead of finding the CO2 normal, we will find it high. So the body is trying to trap more CO2. Of course, if the CO2 is high, this normally should make acidosis. But if it happens with alkalosis, then we know this is respiratory compensation for alkalosis. Let's look into the acidosis here. If it was the pH is low and we look to the PCO2 and we found the PCO2 is pH is 7.4, so it's normal. Yes, it's normal. The compensation is the correction. How the body tries to correct the pH. That's the compensation. So if the pH is 7.4, yeah, that's normal. It should not be anything. But usually there will be a scenario and there will be giving you the levels of the blood gases analysis. So you will be given the pH level, you will be given the PCO2 level and the bicarbonate level. And then based on these three values, you will be asked. Sometimes they will not be giving you only these three, they will give you many more. But you only need to look into these three values in order to give the diagnosis and choose the right option. So let's go into the acidosis side. So as you can see, if the pH is low, lower than 7.4, this means it is acidosis. Then you look first into the PCO2. If the PCO2 is high, then you know it is a respiratory acidosis. What is the cause of respiratory acidosis? Any respiratory disease. Asthma, bronchitis, okay, depression of respiratory center, okay, this can cause an increase in the level of CO2. So what's the compensation going to be? The compensation will be renal. How do we know there is a renal correction or renal compensation? By finding high level of bicarbonates. So the kidney is going to reabsorb more bicarbonates. If the bicarbonate is normal, then we know it is uncompensated. If we find the bicarbonate is high, although it is acidosis, then we know this is a renal compensation for respiratory acidosis. While if the acidosis happens and the CO2 is normal, so with the low bicarbonate, we know it is metabolic type. What is the cause of metabolic acidosis? Diabetic ketoacidosis, any causes of metabolic acidosis due to metabolic problem causing the release of metabolic acids into the circulation, lowering the pH value. So here, it is not respiratory cause. It's a metabolic cause. How do we know? We will find the pH low, we will find the CO2 normal, and the bicarbonate level is low. So if the CO2 is normal, then we know it is uncompensated. If the CO2 is less, then we know that the respiratory system is trying to correct the problem of acidosis by hyperventilating. So in certain people who have metabolic disease, sometimes you will find them in deep coma and they are breathing fast. So why is that? That's because the respiratory system is trying to compensate the metabolic acidosis happening in patients by washing out the CO2 to reduce the pH and bring it back to normal. So in this explanation, it helps you to solve any scenario related to the arterial blood gases analysis and into the acid-base balance. So with that, we reached into the end of the lecture. So we open up Q&A. Anyone has a question, you can ask now. Can we have metabolic acidosis? Metabolic acidosis and renal compensation. Well, according to the previous slide, it is possible. Let's say if we have metabolic acidosis and we have large amount of acids, so there could be a renal compensation. But here in this diagram, we are showing the main player. So there could be a renal compensation, but it is not to a large extent. The main contributor of compensation is going to be the respiratory system, if it is metabolic. But of course, you can imagine, in alkalosis, the kidney will, as we mentioned before, will lose more bicarbonate and will keep more acid. It was in the slide before. While the opposite happens when there is acidosis. So, sorry, there is a long message written by one of you. Some of you ask about the pH, respiratory system, but I cannot see. Regarding the physiology of the respiratory system recording, are you going to share the link? Yeah, it's already being uploaded to the drive that you have. So I have already made the file available. Respiratory anatomy, physiology, GIT system is all with you right now in the drive. It's already there in the OneDrive, yes, that you have shared the link. It's already there. Okay. So, can an acidosis be both metabolic and respiratory? No, it cannot be that way. It is either respiratory or metabolic. That's how the scenario will be, and that's in reality how it's going to be. So that's how I'm giving you the rules. First, look into the pH value, then look to the CO2. If the CO2 helps you, then that's it. If it doesn't, then look to the bicarbonate. Okay? And if the CO2 helps you, then you can look into the bicarbonate to know whether it is compensated or uncompensated. If you are looking into the bicarbonate, then you look into the CO2 afterward to know if it is compensated or by uncompensated. So any question regarding the beginning of today's lecture? Some of you were asking. No, you don't have to know the diseases, but I can just tell you an example of respiratory alkalosis, like what happens in panic, because we breathe very fast and this will wash out acid. Metabolic alkalosis, mainly due to ingestion of high alkali, that causes alkalosis within the body. Acidosis, which is respiratory, mainly due to hypoventilation, while metabolic acidosis is mainly due to the generation of metabolic acids, like in diabetes. Yeah, but you don't have to, yeah. Yes, yes. I have seen one of you who wrote the email of cutting down the reading material for the final. However, that's not possible because you know that preparation of final exam questions takes time. So usually we prepare it much earlier. So we have prepared it from the beginning. Some of the questions were even prepared before the lockdown. So we prepared everything, and we have checked the questions and make sure everything is in order. So that's why it is very difficult now to cut certain material, as the questions are covering most of the topic. Probably we can have a revision that could help you to understand the lecture. Understand what's the way questions are going to be and where to focus more. Probably that could help you more, yeah. Well, distribution is basically according to the teaching load. So I think you can focus more on the lectures which are after the spring break. So those lectures which are especially in the beginning, the introductory lectures, are going to be less important compared to the systems afterwards. I think you need to focus more on the GIT and respiratory system. If I would like to choose those two. Skeletal and muscular system, yes, of course, important. Yeah. Reproductive system, which is going to be next week, there are probably a certain number of questions. But you need to focus on respiratory and gastrointestinal system, muscular system. And muscular system need to focus mainly, about what Dr. Hala has taught you. Yeah. Embryology. I'm not very sure I'm not very sure about it. Maybe during the revision, I will try to check and try to help you see how to focus on which topics within the embryology lecture. We can go back to the questions. I think the same settings is being used. No backtracking, and the time here is going to be 2 hours. Well, you can discuss this with Dr. Hala. Yeah, but usually we are all using these settings. So in human structure and function 1 and 2, which was Dr. Hala, we are using this setting. I'm also using it in my own courses, like in biochemistry, the same setting of preventing backtracking. You have more time instead of, you have 2 hours, so that's a long time. Probably we will see. We will discuss it with Dr. Hala, and then we will let you know. So any more questions related to today's lecture? You're welcome. So try to focus on the renal system, okay? Okay, so I will stop the recording. Thank you very much.